My name is Dennis Peters. I'm the Acting Dean of Engineering and Applied Science here at Memorial University. Thank you all for coming out tonight and supporting our faculty's Speaking of Engineering lecture series. I'd like to extend a special greetings to our guest speaker this evening, Dr. Bruce Pike. And I'd also like to acknowledge Mr. Jeff Emberley, CEO and Registrar of the Professional Engineers and Geoscientists of Newfoundland and Labrador, also known as Paganel. Mr. Emberley will come forward a little later to talk about Paganel's sponsorship of this lecture series. Before we begin, I'd like to take a moment to acquaint you with the emergency procedures for this space. In the event of an emergency, please exit the building in an orderly manner. There are 2 exits at the back of the theater where you've come in, and one here at the front. Please exit through the door closest to you. All 3 exits take you into the lobby, of course, out through the front door, and turn left. That's right. And the muster station is in lot 22 on the east side of the building. The east side of the business building. More likely to be useful, the washroom facilities. Once again, out through the doors at the back, and just to your left here, there's male and female washrooms. I don't know, there was some difficulty with the light in the female washroom earlier, so if the light doesn't come on for you, I'm not sure if that's been fixed. If that doesn't work for you, turn right, and it's your down the hallway, the second hallway on your left, on this side, moving into the computer science wing over here. The Speaking of Engineering lecture series promotes engineering in our province and raises awareness of engineering-related issues among students, the academic community, and the general public, which we feel is very important. The lecture series is an initiative of Memorial's Faculty of Engineering and Applied Science and is co-sponsored by the Professional Engineers and Geoscientists of Newfoundland and Labrador. Paganel is an invaluable asset to our program and our graduates, and we are honored to partner with them to bring you this lecture series. Tonight's lecture is about how engineering has revolutionized brain imaging. Discoveries and advances in engineering and physics have revolutionized medicine over the past century, and nowhere is this more apparent than in brain imaging. Technologies such as X-ray, CT, or X-ray CT, CAT scanning, magnetic resonance imaging, also known as MRI, positron emission tomography, or PET, and another one that I'm not going to pronounce, or MEG, have opened. I'll ask Dr. Bruce Dr. Pike to pronounce that one later for me. Have opened unprecedented new windows on the living human brain. Tonight, Dr. Pike, head of image science at the University of Calgary, will review some of the major developments in this field and share his thoughts on what the future may hold. I'm proud to say that Dr. Pike is a graduate of Memorial. He obtained his Bachelor of Engineering degree in Electrical Engineering here, and then attended McGill University, where he obtained his master's and doctorate degrees in Electrical Engineering, working on magnetic resonance imaging technology. He then conducted postdoctoral studies in radiological sciences at Stanford University. From 1999 to 2013, he was director of the McConnell Brain Imaging Center at the Montreal Neurological Institute of McGill, was a Killam Professor of Neurology and Neurosurgery, the James McGill Professor of Biomedical Engineering, and a Professor of Electrical Engineering, Physics, Medical Physics, and Radiology. In 2014, he joined the University of Calgary as the Campus Alberta Innovation Program Chair in Healthy Brain Aging, Head of Image Science, and Professor of Radiology, Clinical Neuroscience, and Biomedical Engineering. Dr. Pike has published over 300 scientific papers and book chapters that have been cited over 23,000 times. He has also been featured on the cover of the scientific journal Nature. He is an editor of the top-ranked brain imaging journal, NeuroImage, a senior fellow of the International Society for Magnetic Resonance in Medicine, and serves on the advisory boards for a long list of national and international organizations. Please join me in welcoming Dr. Pike.
Thank you very much for the introduction. I've got a microphone on here, and I can't really tell whether you can hear me way up back, but you can? Good. Well, let me get started. Start with a little outline of what I planned on talking about today. I might just use the mouse down here to point on things. What I wanted to do really was start with just a very general talk, hopefully that's approachable for everyone here. It starts with a little bit of history about brain imaging, and I'm going to talk about it mostly from a brain imaging research perspective rather than radiological clinical diagnosis perspective. I'm going to try and go over some of the major imaging technologies that we use today and the engineering and physics basis behind them, with a focus on magnetic resonance in particular, where I'll try and highlight some of the major engineering innovations that have occurred over the past now 40 years or so that have really revolutionized what neuroscience we can do. And at the end, I'm going to try and finish off talking a little bit about where I see the future going. And as was mentioned, I actually did my undergraduate degree here at Memorial, graduated in 1984, and it was right around then, I guess I was doing a work term, an academic work term at the Health Science Center, and they were installing the first X-ray CT in Newfoundland. And there was a commissioning process, and I became involved in that and became quite intrigued by that. And these were still pretty early days. And from there, I kind of looked around to see where I could work on medical imaging, and it turned out that the Montreal Neurological Institute of McGill was a real international hub for brain imaging research, and I made the move there to start graduate school and worked on the very first superconducting MRI scanner installed in the country, which was also at the MNI, that physically sits in the same location as the existing 3T in the McConnell Brain Imaging Center. But the story of brain imaging really, you can argue when to start, but a good place to start always is with Wilhelm Röntgen, who in 1895 was the first to produce and detect electromagnetic radiation in the X-ray range. And he actually won the very first Nobel Prize for physics that was given in 1901. I'm going to try and highlight, as I go through the talk, actually a series of Nobel-worthy contributions to the field of brain imaging, and certainly Röntgen was the first. So, you could really look and say that was the birth. So X-rays and brain imaging, if you just took even a good quality lateral projection X-ray of a whole head, you might see something that looks like this. It gives a very nice view of the dense bone. You're essentially seeing a shadow, effectively showing the X-ray attenuation map as a projection image such as this, but as far as the brain goes, this is great for the skull and teeth and so on, but in terms of actually looking at brain tissue, it's quite poor. And there were a bunch of methods that were introduced involving some things like injecting air into the ventricles of the brain, trying to improve visualization of certain aspects. But in general, X-ray projection imaging such as that is a very poor window into the brain. One of the things that came along, I mentioned, was introducing contrast agents. And the image you see down here is what's called an X-ray angiogram. Again, it's a projection similar to this one, a sort of lateral left-right projection. But in this case, a series of images were taken. One that looked much like this, and then another one where there was an injection of an iodinated contrast agent, iodine-based contrast agent, into the carotid artery that you see coming up here. And as that contrast agent fills the blood vessels, it increases the attenuation of X-rays there. And if you do a subtraction of the image without the contrast agent and with the contrast agent, you can remove the signal from everything except those blood vessels. And you can produce some quite spectacular images of the cerebrovascular system. And this is still used quite extensively as really a gold standard way of looking at blood vessels in the brain, and similarly in the heart and other areas. But again, we're not getting at brain tissue itself. So the really next big thing that I mentioned, already is X-ray computed tomography, so CAT scanning, it's also known, computerized axial tomography. And if you sort of look at the history of CT scanning, it's really intimately linked to the development of computing power. Some of the theory behind it really existed for a very long time. Some of the fundamental mathematics developed a century ago by Radon and others. But the actual practical implementation really required the development of computing power. And what you see on the bottom here is actually an X-ray CT scanner with the covers taken off. And you can see just a little illustration with the green arrow pointing to the X-ray source on this side and a range of detectors on this side. And in this configuration, it's sending sort of a fan beam of X-rays. And sort of conceptually, the idea is that if you place a head inside of that opening and you rotate this apparatus around and get projections, much like the one we saw before, but if you get that from many, many directions, you have a mathematical problem of trying to say, well, what must the object be such that these are its projections from all those angles? And by doing that, you can produce images of the brain which look something like this. And this is a modern X-ray CT image. It certainly didn't look anything like this when it was first introduced. But as you can see, again, it's X-ray based. It's showing a map of X-ray attenuation coefficients. And the skull is showing up very bright, high attenuation. And the difference between the white matter, shown a bit darker here, and the cortical gray matter, shown a little bit brighter, is subtle but detectable and extremely valuable clinically. This is now a bread and butter tool, as we know, in diagnostic radiology. And the development of X-ray CT you can trace largely to Sir Godfrey Hounsfield. And I highlight him because, first of all, he was, I believe, the first electrical engineer who was given a Nobel Prize in medicine or physiology. And he did this work, was awarded this in 1979, but Godfrey had worked in a company in Great Britain on some of the first commercial all-transistor computers. And I had mentioned that CT was intimately linked to the development of computers. So in 1958, it was the EMIDAC 1100 was sort of early computing power. And during the 1960s, he worked on this problem of trying to determine what was inside an object by examining its projections from multiple angles. And he worked for a company, EMI, as in the EMI DEC, and as many of you may recollect, EMI held Apple records and the rights to the Beatles. And the story goes that they actually made substantial profits from the Beatles during that period, and they actually used much of their investment in some of their basic research labs to fund Sir Godfrey Hounsfield's research. So in 1971, they actually scanned the very first patient. And I don't have the first X-ray CT image, but it was atrocious by today's standards, of course. Just a footnote, since in particular, since I just met a South African colleague, Hounsfield shared the Nobel Prize with McCormick, who brought a lot of the mathematical work. And I believe McCormick was South African. I don't know, you can correct me if I'm wrong. PET and SPECT. So two other major technologies that are now mainstays in clinical medicine and in neuroscience research are positron emission tomography and single photon emission computed tomography, shortened to PET and SPECT. In positron emission tomography, the only real difference between these and X-ray CT conceptually is that in X-ray CT you have an X-ray source outside that you project through the body and detect on the other side, whereas in these nuclear medicine methods you actually introduce radioactive material into the body and you have detectors surrounding it which can detect the emitted radiation. So there's not a source outside and a detector on the other side, but there's the radioactive material inside with surrounding detectors. And that's sort of the cover of a modern head-only PET scanner, where you see this whole ring of detectors around here. And in the case of positron emission tomography, the idea is you have a radioisotope that decays by the emission of a positron. A positron is a positively charged electron. It's the antimatter of an electron. So as soon as it's emitted, it immediately is attracted to a surrounding electron. So positron and electron collide and they annihilate and produce energy that's always the same, exact same energy, 511 kiloelectron volt gamma rays that are emitted, which is given by Einstein's equation about the relationship between energy and mass. So you get these two gamma rays that shoot out, and they're detected simultaneously by these detectors. So if an event is detected here and here, you infer that there must have been a emission of a positron somewhere along that line. And by doing that continuously over a period of time, you can build up a map of that distribution. And this is typical of what you might see in a PET scan here. And the images are generally lower resolution and show less anatomical detail than X-ray CT or MRI, but the interesting part is that that radioactive material that you introduce can be tagged to an interesting molecule. So a very common one that's used is a glucose analog. In case of most of the oncology imaging that goes on in the world uses a tracer that's based on fluorine 18, labeled with a glucose, so it's called the fluorideoxyglucose. When you ingest this, it produces a map of glucose utilization. And glucose is one of the substrates of metabolism in the body. And if you have areas such as malignancies, metastasis, so on, that are utilizing a lot of sugar, a lot of glucose because they have high metabolism, then you'll get a large concentration of that tracer there. So they can produce, in that case, a map of metabolism. But you can actually take these radioactive isotopes and tag them to all kinds of other interesting molecules to look at distributions of all kinds of neurochemicals in the brain. So that's an extremely powerful technique, but is complicated and expensive to produce some of the tracers, the radio tracers. The other one that was mentioned in the introduction is called magnetoencephalography, or MEG. And it's similar conceptually to electroencephalography, which is EEG. And many of you have probably heard of EEG or ECG, electrocardiography. ECG, you're putting electrodes on the skin, on the body, and detecting electrical activity associated with the heart. In EEG, electroencephalography, you're putting electrodes on the scalp and detecting electrical activity associated with the brain. And in magnetoencephalography, you have a large detector. It looks like an old-fashioned beauty parlor hairdryer. That's a couple of $1 million. And it actually detects magnetic fields associated with electrical currents in your brain. So every time there's an electrical current, we all learn in engineering that if you have a current flowing in the direction of your thumb in a wire, you have a magnetic field that wraps around in the direction of your fingers. So all those electrical currents that are occurring in the brain are generating magnetic fields. And this device is the way to detect those. So it's extremely powerful tool. It detects extremely small natural magnetic fields down in what's called the femtotesla range. So you'll hear me talk about MRI scanners a bit later, and I'll talk about 1.5 and 3 Tesla magnets. Those are the typical field strengths, which are already some 50 to 150,000 times stronger than the Earth's magnetic field. But these are now, billionths of a fraction of the Earth's magnetic field. So they're extremely tiny. But they've really been enabled by a whole series of innovations related to superconductivity, because the detectors that are used here need to be superconducting. But they allow you to monitor in real time, so very fast on the millisecond time scale, activity, magnetic field activity inside the brain of an awake behaving subject. So they've become very powerful tools for neuroscience research and for some clinical activity as well. But I wanted to spend most of the time talking about magnetic resonance imaging. And magnetic resonance imaging, I'll give a little bit of background on. It's based on this phenomenon called nuclear magnetic resonance. That's been around for 80, 90 years or so. And it's really a mainstay of, chemistry and physical chemistry, where you want to study the structure of molecules. And it's it's a phenomena that uses strong static magnetic fields and radio frequency electromagnetic fields. So static magnetic fields like the Earth's that we live in all the time, radio field like the ones you're transmitting from a radio tower or from your cell phone. And it's in fact those kind of features, the static magnetic fields and the radio frequency fields, which cause us to be confident in its safety. So unlike ionizing radiation, it has no known harmful biological effects. So that's valuable for all medical purposes, of course. But from a research point of view, it also makes it a ethically acceptable tool, for example, to repeatedly study healthy subjects. If you want to look at the brain development in a group of children from birth to high school, and you want to scan them every 6 months, it's perfectly ethically acceptable to do that because it's not harmful. It would be unethical to do PET scans on them every 6 months for that period, or to do X-ray CTs, CAT scanning. So it's considered an extremely safe technology, and it provides exquisite soft tissue contrast. So not good for looking at bone and other things particularly, but extremely good at looking at soft tissues, such as the brain. And it's been sort of continuously evolving, rapidly evolving technology. And one of the things that makes it so interesting and powerful is, unlike something like X-ray CT, where you are forever just generating maps of X-ray attenuation coefficients. With MRI, you can actually look at brain structure, brain function, physiology, chemistry, and so on. It's a much more flexible tool. And really, I'll try and do this one quickly, but this is just a sort of a brief list of some of the key Nobel Prizes that have been awarded to the development of nuclear magnetic resonance and magnetic resonance imaging over the years. And it goes back to Rabi in 1944, Physics Prize, and actually the International Society of Magnetic Resonance Imaging continues to name its Young Investigator Award every year after Isidore Rabi. But Bloch and Purcell shared the Nobel Prize in 1952 for their development of NMR. And I actually bumped into Felix's son at a cocktail party once. That's one of my brushes with fame science geek stories. Richard Ernst came to the opening of our lab at Stanford. He received the prize in 1991, related to NMR primarily in chemistry. And the two other, there's Kurt in 2002, but Paul Lauterbur and Peter Mansfield were two mainstays, regular attendees at our magnetic resonance and medicine meetings for years and years, and they shared the Nobel Prize in 2006 for the development of magnetic resonance imaging per se. And there's a couple of interesting little footnotes in there. One, Paul Lauterbur, who really submitted the first paper ever describing magnetic resonance imaging to Nature, had it rejected with the editors claiming this would be of little interest to anybody. But it was later published and is now considered one of the landmarks publications in that entire field. So, a very brief bit of the science. So, nuclear magnetic resonance is a phenomenon of the nuclei. And any nuclei which has an odd number of nucleons, or neutrons and or protons, has something called a magnetic moment or spin. And in biological tissue, the most prevalent one that we look at is hydrogen nuclei associated with water. So it's in great abundance in the body. We're made up of a large amount of water. And the nuclei in there are just a single proton. And those single protons have a charge, positive charge, and they have an inherent spin. So you've got a spinning charge, and again, we know if we have current going in a loop this way, we're getting a magnetic field in the direction of our thumb. So those spinning nuclei have the property of looking like a little bar magnet, a little north-south bar magnet. So all the protons in your body are behaving this way. And it turns out when you take them and you place them in a magnetic field, there's an interaction called precessional motion, whereby that spin wobbles around. Just like if you take a spinning top, a child's top, and you spin it in the Earth's gravitational field, you place it vertical on the table, it'll just spin on its axis. But if you place it at an angle, it spins on its axis, but it also wobbles. That's called a gyroscopic motion or precessional motion. It turns out that wobbling, how fast it wobbles, depends on how big it is, how fast you spun it, but also depends on the strength of gravity. So if you have the same top spinning at the same rate, it will spin differently depending on the gravitational field, directly proportional. So if you take it on a spaceship in outer space and you spin that top, of course it doesn't matter which angle you put it at, it won't precess, it doesn't wobble. So that's called the Larmor equation. It just says that the frequency at which it precesses is a characteristic of the nuclei times the strength of the magnetic field. So why is that interesting? This is an equation established by Joseph Larmor. Well, that's interesting because for hydrogen nuclei, that constant of proportionality is about 42 megahertz per Tesla. So if you take a glass of water or a person and you put it in a 1 Tesla magnet, and the hydrogen nuclei in there are going to precess or wobble at 42 million times per second. So that is right in there on our FM dial, right? So I'm not sure, I can't remember what CBC, CBC is AM here still, isn't it? Yeah. So at FM 97, something like that, that's 97 megahertz. So if you had a 2 Tesla magnet, you'd be right up there in 85 megahertz. So a lot of the technology surrounding MRI, both NMR and MRI, really involve transmission of radio frequency waves. They involve generating big magnetic fields, and they involve transmission of radio frequency pulses using antenna designs and so on in the near field, and then antenna designs to receive back radio frequency signals which are emitted from your body. So you can actually listen in to those wobbling nuclei in your body. I mentioned Tesla, and Tesla is the international scientific unit for magnetic flux density. So you'll hear me talk about this. But just as a footnote, Tesla is actually named after Nikola Tesla, who was a Serbian electrical engineer and inventor, and was a contemporary of Thomas Edison, and actually worked for Thomas Edison for a period. But they had a very strong falling out. You can trace the development of alternating current, the fact that we use AC current coming out of our walls instead of DC current as Nikola Tesla, AC motors, X-ray tubes, there's sort of a huge list of patents and discoveries by Nikola Tesla. And him and Edison despised each other to the point actually that they both, in the early 1900s, stated openly and publicly that they would refuse a Nobel Prize if the other one was awarded it as well. And it turns out neither one of them ever was awarded the Nobel Prize. But he's also, if you're ever looking through real biographies, I mean, he's the quintessential mad scientist. He ended his life talking to pigeons in Central Park and in poverty. He's an odd-looking in his picture there. But there's also a movie called The Prestige. I don't know how many of you have seen the movie The Prestige, but the character of Nikola Tesla appears in The Prestige and is played by the now late David Bowie. So I thought I'd throw that in as a little footnote. So back to NMR. So the idea with these nuclei is that we can disturb them from an equilibrium state. And if we do that and we have a little receiving antenna, a little coil of wire next to your head, then we can actually pick up these wobbling magnets, these wobbling nuclei, and they appear as a little radio frequency signal that we can detect, very, very small. but that's essentially what we're doing with an MRI scanner. And if we calculate the spectrum of that, we see a little peak associated with water. And that peak occurs, the frequency of that will actually tell us the strength of the magnetic field we're in. So that's handy. So imaging, as I mentioned, was Paul Lauterbur. He's the guy who, so that was the basis of nuclear magnetic resonance for studying chemistry, chemical properties of materials for years, decades, still is. But Paul Lauterbur came along and figured out a way to actually generate images from this. And the key there was if you had a whole object, like a head or a whole body inside of the magnet, it wasn't good enough to figure out what the total signal being transmitted from the head is. You needed to figure out how much signal is being transmitted from each spot, so to speak, inside of an image. And he figured this out by recognizing this behavior of the frequency of the signal being transmitted being related to the strength of the magnetic field. So he introduced a magnetic field that varied in strength linearly as a function of position. So for example, from head to toe. So your overall magnet might be 1 Tesla, but if he introduced a variation in the strength that he could turn on and off between 0.9 and 1.1 Tesla, and then he received the signal from everything, he could figure out that this frequency must be coming from this part of the body. And that was kind of the fundamental key idea that he introduced that allowed him to produce images and to later be recognized by the Nobel Prize. This was the footnote about his submission to Nature. So, this is kind of typical of the kinds of images that you can produce routinely with magnetic resonance imaging now. This is just a single slice through a head this way, what we call the sagittal plane. But what I hope it conveys is that it shows very nice detail. This would be a typical clinical quality image. You can easily distinguish white matter and gray matter, and the ventricles filled with cerebrospinal fluid, as well as muscle, and you see bone is generally dark in here. You can see some of the marrow in the bone in the skull. But it produces exquisite images very quickly, and from a clinical diagnostic point of view is now the go-to method for looking at many neurological potential conditions. Just for the engineers out there, this is essentially the raw data signal that we receive. The way we collect data in MRI is the raw signal that comes out of our receive coils following demodulation is in Fourier space. So the reconstruction process is a simple multidimensional Fourier transform. So for those of you not engineers, you can ignore that part. But there's a very elegant Fourier description of all of this, which allows the design actually of sort of ever more fascinating data acquisition strategies. So one aside, another sort of amusing, I think, footnote to MRI and Nobel is Ray Damadian. So Ray Damadian was another character who was always at our MRI meetings for years and years. Excellent scientist who did a lot of pioneering work in the field. But he was not, when they awarded the Nobel Prize for MRI to Paul Lauterbur and Peter Mansfield, they didn't include Ray Damadian. And for good reasons, I think, widely accepted. But this guy is a character, and he was so offended that he actually took out full-page ads. And this is a copy of full-page ads in the New York Times and the LA Times on October 9th and 10th of 2013 with a picture of the Nobel Prize upside down, demanding that this shameful wrong should be righted. And he actually went on a huge campaign. So if you're ever looking for a story about how to not win a Nobel Prize gracefully, then you should look up the story of Ray Damadian. So I put up this image, which has a little movie playing over here. And I put this up because the other thing, I hope I've touched a little bit on how you can produce exquisite images of individuals and use it to diagnose brain tumors and multiple sclerosis and all kinds of other things. But the safety of MRI and the high quality of the structural images have allowed us to do an amazing amount of neuroscience research that wouldn't be possible otherwise. And I've put up a couple of examples here, but these have also really only become possible with current sort of engineering and computer science methods that allow us to process vast amounts of data. So on the left-hand side here is some data that we collected in a large NIH study, National Institutes of Health study, looking at the development of the healthy brain. So as many of you may remember, if you had little children, when you go to your pediatrician, they might weigh them, they might measure their length, and they might measure their circumference of their skull, and they might show them to you on a chart and say, oh, they're in the 48th percentile or the 25th percentile or the 70th percentile, whatever. There was some normative data about that. What NIH wanted in this study was for us to establish normative data about how the brain develops. So we actually looked at 500 children longitudinally over 8 years. These are healthy children sampled across the socioeconomic spectrum and so on. And we did behavioral testing and cognitive testing and all kinds of neuropsychological evaluations every time, and we scanned them on a regular basis. And this is the group of children between birth and 4.5 years of age. And I show that, but it's also all of them now taken and mapped into a common space, so non-linearly transformed, warped until they align and match, so you can remove gross shape effects and so on. And now just look at the average of all of those and how it evolves. So you can see, I put this there for this because there's dramatic changes which happen in the first year of life. There's this beautiful kind of flowering, if you want, of the brain as you get myelin sort of insulating coating surrounding the axons that spreads through the brain during those first years of life. So, in addition to that sort of routine diagnostic purposes, there's this vast amount of research questions you can do. And then on the other end of the age spectrum, perhaps more relevant for old guys like me, is the aging brain. And again, this is a large population data set from groups of individuals ranging from normal controls to those with mild cognitive impairment, all the way up to early Alzheimer's disease. And by taking data from hundreds of subjects and looking at these categories, you can do a statistical analysis to say, for example, what areas of the brain are losing volume relative to those healthy controls as a function of the extent of dementia. And that's what's shown in those color maps on top. So MRI is now turned into this extremely powerful tool for studying brain structure. And that's kind of the main point of that. Scanner hardware, I already talked a little bit about this. I don't need to dwell on it too much, but the main components are this large magnetic field in Tesla, these gradients which allow us to do imaging, and radio frequency coils or antenna which allow us to transmit and receive signals. This is what a magnet looks like if you tear off all the plastic covers. They're superconducting solenoid designs by and large. There are some differences, but the most common ones you'll see are basically big toilet paper roll with wire wrapped around it. The wire is typically a niobium titanium alloy that's many kilometers long, and it's immersed in liquid helium, which has a boiling point of minus 269 Celsius. And at that temperature, the wire becomes superconducting, meaning the resistance vanishes to exactly zero. And large DC currents are injected into those on the scale of hundreds and hundreds of amperes are flowing nonstop in those coils without a power source. So basically you get the current going and you remove the source, and the current flows indefinitely because there's zero resistance. So these superconducting magnets have very high magnetic fields. They're on all the time. The biggest danger associated with them is actually getting near them with a piece of iron if someone's in the magnet, because they'll be attracted with such force that they could kill you. Sort of to date, the range of magnetic fields that are being used in imaging are maybe less than 0.05, but maybe 0.1, all the way up to the highest, I think, is 12 Tesla that's ever been built that could put a human in. The other is that they have to be very uniform, typically a uniformity over the size of the head of about 1 part per million. And those are the contributing things that make them expensive. So the Beer magnets, these systems are, the rule of thumb is about $1 million a Tesla. So if you want a 3 Tesla magnet, they're around 3 million. A 1.5 is around 1.5. They tend to go up a bit non-linearly after that, probably 10 million for a 7 Tesla. These coils that produce the variation in the strength of the magnetic field these are basically huge extra windings that go in there that are not superconducting. So these are resistive, and they're embedded in like an epoxy resin. You can just see some of the wires going through there. And in this case, they're varying in the density of the winding, such that the strength is stronger going down towards 0, and then stronger in the negative direction there. That's how you produce that linear variation. They're loaded inside the bore of the magnet, and they're driven by, you see these huge big cables here. So those are resistive. You've got massive amplifiers running at rail voltages over 1 kilovolt that are dumping huge currents down there. And they're getting hot, and now the latest systems have lots of plumbing in them to try and water cool them. So there's been, for a long time, that was a big technical limitation. And engineering innovations pushed those to enable faster and faster scanning. And that was kind of, for many years, a big bottleneck. And really, the limitation now is that if you switch them too quickly, you'll induce currents in the body which cause twitching of the muscles. And so there's limits on how fast you can turn them on and off, lest you, God forbid, induce cardiac arrest. And the gradient system innovations, the other person who shared the Nobel Prize was Peter Mansfield. And a lot of the gradient innovation design goes to Peter Mansfield, and Peter just died earlier this year, but was a real figure at our meetings. The other is antennas. I mentioned how we transmit and receive radio frequencies. This is kind of typical if you're looking at the opening of a magnet and a couch where a patient might lie. Their head would typically go inside of this, and this is a radio frequency antenna. And that it turns out that antenna design, when you're near a head, coil design is best. And they're detecting extremely small magnetic fields in here. And you generally have these receiving antenna matched to the object you're imaging. So if it's a head, it looks something like this. Or this. If you're imaging a wrist, you would have a little antenna which fits right around your wrist. Similarly for breasts, or you might have ones that can wrap around the shoulder or so on. But these antennas, there's a lot of antenna design that's taken place. And one of the recent technical waves has been to introduce arrays of coils, where you don't just have a single antenna, but you have whole arrays of them, such that you benefit from the increased sensitivity of a small coil, but the ability to cover a large area by having whole arrays of them. That allows you to speed up acquisitions quite a bit as well. So I'm going to move on to talk a little bit about functional imaging, because I've focused on structural right now, but the other really big innovation that's happened has been functional imaging. And this is a picture of a very famous Canadian neurosurgeon that you've probably seen those heritage moments on television about, Wilder Penfield. Wilder Penfield was the founder of the Montreal Neurological Institute back in 1936. Here he is standing at the board, drawing the brain. And Penfield is responsible for a lot of the early work in terms of mapping out the functional organization of the brain. So what areas of the brain are involved and are responsible for different aspects of our function. And the way he did a lot of that was during open craniotomies, where a large piece of the skull is removed, on awake patients. So these were mostly epilepsy patients, but some others as well. And they were undergoing surgery, and he would remove a large thing of the skull under local anesthetic. The patient was awake, and he was able to directly stimulate the cortex with an electrode and see the responses in the awake patients. And you can see he's actually placed all these little sticky labels on the surface of the brain exposed here, and took meticulous notes about the responses of these patients. And he's responsible for this now extremely famous drawing, which shows a cross-section of the brain cut this way, and on it he's drawn out and labeled areas of the brain that are responsible for motor and sensory features, of the toes all the way up to the knees, the hands, the mouth, the tongue, and so on. And actually there are these two beautiful and grotesque things called the sensory and motor homunculus, where these are little clay figurines where the size of the body part has been scaled based upon the cortical representation behind it. So as you can see, the hand areas of the motor cortex are much, much larger than the calf. So this is the motor homunculus, the sensory. But that's how we learned largely about functional brain organization. But around 20 years ago now, maybe 22, work by Sergio Gala developed a technique which is now a mainstay of neuroscience research called functional MRI. And that indirectly detects neuronal activity in the brain by looking at something called blood oxygenation level dependent effects, or BOLD effects. And it comes from the magnetic properties of hemoglobin. Hemoglobin is the protein in red blood cells that transports oxygen. We can now acquire structural images such as that in gray. In this case also shows in red a tumor, and areas associated with the hand and face sensory areas that are stimulated, that were investigated prior to surgery, in this case, to look at the proximity to the tumor that was going to be resected. But the phenomena comes about by a discovery of Linus Pauling. So Linus Pauling, many of you probably remember, who was a two-time Nobel laureate, one for chemistry and in 1962 for his anti-nuclear peace efforts, a Nobel Prize for Peace. But basically, depending on whether oxygen is attached to hemoglobin or not, it actually changes from what's called diamagnetic to paramagnetic. It changes its magnetic properties a small amount, and that can be detected with MRI. So a typical kind of fMRI experiment now, and this is a very simple one, is you run your MRI scanner like a movie camera, just constantly imaging using very fast imaging techniques introduced by Mansfield and others. So maybe as short as every 50 milliseconds, but typically you'll get the whole head at every second or two. So you get a constant movie of someone lying in the scanner, and you've acquired the data to be very sensitive to these changes in the hemoglobin. And then you might have them perform a task for a period of time and then rest, and perform it again and then rest, or visualize a stimulus or what have you. In this case, it's a visual stimulus. Then when you take that whole time series of data, you can ask a statistical question, which is, what areas of that whole brain varied in synchrony, highly correlated with the task or the stimulus you gave, and produce a statistical map shown in color in this case, overlaid on top of the structure. So in this case, you're seeing the primary visual cortex, which we know sits in the back of the occipital lobe. So this is now a way to do functional mapping in awake subjects. And I'd like to put this one up because this is actually a data set collected in my lab in around 1999. So already getting almost 20 years old. that shows a subject was lying in the scanner. They were looking at this cross sign here, and there's this checkerboard flashing on and off that spins around. And if you do an analysis of that data, this is just a slice through the brain this way that cuts right through the back of the head, back where we know the visual cortex is, you can actually see the response in the brain in real time to that stimulus. If you stop that somewhere, like there, you can actually see this. This is a sort of a classic neuroscience experiment because it shows what's called retinotopic mapping, whereby when you look out into the visual world, everything in this hemisphere of your visual field is represented in this hemisphere, the occipital lobe, and vice versa, and that there's a one-to-one mapping whereby two adjacent points in your visual field are actually represented by two adjacent points on the brain. And this was originally established through meticulous experiments in animal models with radioactive materials, where you would stimulate them and sacrifice them and study the distributions. And now it's like a parlor game trick that you can basically do. But in addition to those kinds of experiments, we also do experiments now called resting state fMRI, where you can just have someone, instead of doing a task, they simply lie inside a scanner while you scan them, and you can study natural fluctuations in the response in order to tease out different brain networks. And there's now sort of a whole series of well-established brain networks because we know it's the brain is a whole wired apparatus. So these kinds of experiments have sort of changed the way we look at the brain substantially, both in health and in disease, where we might look in certain diseases at abnormal functioning of whole brain networks. I was going to mention diffusion a little bit, but I'm going to skip that for a moment because I'm running a bit late, and talk a little bit about what's on the horizon very quickly. So one is going to ultra-high magnetic field. So the latest emerging research magnetic field strength is 7 Tesla. And I put this up because this image here, anyone who is used to looking at MRIs knows this is a gorgeous, beautiful MRI, shows beautiful detail of the brain. And it actually is a post-mortem brain. So this is a brain from cadaver that was removed and fixed in formalin and scanned for 36 hours on a 3 Tesla scanner in Montreal. And we were using this was from an MS patient, and we were using it to study subtle cortical pathology in multiple sclerosis. That's almost impossible to see on clinical scanners. But this is actually a blow-up of a very small area of cortex, just a tiny bit, on a patient acquired on a 7 Tesla scanner in a 10-minute scan that actually shows beautifully this little tiny area of abnormal pathology in the cortex of this MS patient. So ultra-high magnetic fields are going to change what we can see in the brain by really zooming in to another level of detail. That's true also of MRI, and this is again a little bit of a party trick, but it gives you some sense of that retinotopic organization again when you can start to resolve at a very fine scale. So this is a subject lying inside a 7 Tesla MRI scanner looking at the letter M on the screen. And over here we see their brain. Because the brain is all folded with sulci, it's inflated, so blown up mathematically. It's blown up such that it's flat like a balloon. And then about 2 millimeters is peeled away. So you're looking about 2 millimeters deep into the cortical layer. And you can see the letter M on the cortex of the brain. So that's the kind of detail we expect to get to. So the other I'll allude to was looking at white matter fiber tracks. So in the brain, axons are connecting all different areas of the brain, and until recently we've really had no way to follow those and figure out how things are connected other than through post-mortem analysis. So, and this is some work from Leipzig, one of the Max Planck Institutes in Germany, that actually shows now, again, and this is This is the ventricle in here, so maybe this is 2 centimeters by 4 centimeters, a very small area. But it allows us to actually go in, look at the microscopic diffusion of water molecules and trace these white matter tracks, these connectivity, at a level that's unprecedented. PET and MRI are being integrated simultaneously in scanners where we can look at brain structure in great detail as well as brain chemistry. And then the very last thing I'm going to finish on is something, a new program that I've just launched in Calgary. And we just found out this past week that we received an $8 million grant from the CFI for this program, is the integration of focused ultrasound and MRI. And the idea in this case is to use a large helmet-like device that goes inside the MRI scanner and can introduce high-intensity ultrasound to the point that you can actually heat and destroy tissue. And the idea with that is you can focus it on very small areas of the brain to do neurosurgery. And I put in here the last thing that I'm going to show, which is just a little 3-minute video. I put it in because it actually shows the treatment of one of the first essential tremor cases that we did.
So essential tremor is a neurological disorder. It's very common. It's the commonest of all the movement disorders. Not that many people have it so severely that it disables them from doing everything. It means that somebody can't sign their name. It means that they can't bring a cup of coffee to their mouths. Elias has a severe form of essential tremor and is disabled by it, and that's why he was referred for the high-intensity focused ultrasound treatment. On the morning of the procedure, he comes in with his hair shaved, but then we repeat that shave after I've given him freezing all the way around his head. Then I put the frame on his head with 4 pins as low as possible, and then essentially a very tight rubber holder goes around his head, which will then allow the water to completely bathe his head, because ultrasound, of course, can't go through air, it has to go through the water. Then he goes into the MRI machine and spends 4 to 6 hours in there while we do imaging. And then we start making test lesions where we ask him whether or not he's feeling something when we apply, as we increase the intensity of the ultrasonications. So Dr. Bruce Pike was recruited to Calgary a number of years ago, and he is one of the world leaders in MR imaging. He certainly initiated this process of bringing high-intensity focused ultrasound to Calgary.
It's very intriguing. Obviously, the idea of neurosurgery in an awake patient without breaking the skin is revolutionary. Getting through the skull in particular is one of the most difficult areas to get to with ultrasound. So the procedure is really quite interesting in that we acquire essentially routine structural images, and then we apply the focused ultrasound first in lower intensities, so we're able to heat it just a few degrees above normal temperature, and while we're doing that, we actually produce images that are temperature maps, and so we can see exactly where we're heating. And it turns out that heating, if you heat to a high level, you'll burn and destroy the brain tissue. But if you heat to lower temperatures, you can actually produce a temporary physiological effect. So not only can you produce the temperature maps and see exactly where you're heating, you can actually interact with the awake patient and see the physiological response associated with changing that precise bit of tissue. In my own vision, this is sort of the beginning of a much larger research platform. So there are some things that we begin with immediately, such as essential tremor in terms of treating, and patients will benefit immediately. And then to start investigating other potential treatments, and really to see them come in and sort of lie in the MRI scanner and 2 hours later get up and walk out and take a drink of water and sign their name it's impressive. So it really is life-changing for them. And yeah, that's extremely gratifying.
that's just a video that was made for a press release for one of the early cases we did over the summer. And I will stop there and really, just conclude that there really has been this revolution that's happened in neuroimaging. It's been driven by engineering and physics and the fields of neuroscience and engineering and physics are sort of inseparable now, really. They're intimately linked, and I don't think anyone in any field of neuroscience are you going to compete internationally if you're not sort of intimately involved with pushing technologies forward. Yeah, I see this not stopping anytime soon. So thanks to everyone who's funded me and worked with me for the years. One of my PhD students, Matthew, is here with me today. He doesn't follow me around for talks. He lives here now. But yeah, it's a pleasure, and I'd be happy to answer any questions. And sorry for going over a bit in time. Thank you.
Thank you very much. We will entertain questions if there are any.
Yes. Yes.
Where exactly applied outside of the brain?
Yeah.
So, I mean, applying that in the brain of any organ system would be probably the most risky endeavor. So where has that been applied in the brain?
So yeah, it's been around for a number of years in treating uterine fibroids and also in bone metastasis. But getting it through the skull is the hard part, right? It's very difficult to do that. So it's really been achieved with these array of 1,024 focused ultrasound transducers where you can do some electronic steering and compensation. So you actually need to acquire an X-ray CT image of the skull and build a model of its effects and pre-compensate for it to do it, and also to be able to monitor in real time. But there's sort of two other really exciting things about the focused ultrasound that I lead to in this platform. One is using it in combination with the injection of microbubbles. You can transiently and focally open the blood-brain barrier for drug delivery. Okay? And it's also been shown in animal models to open the blood-brain barrier for removal of beta amyloids plaques in dementia models. And there's clinical trials going on in gliomas and in Alzheimer's already. And the other is there was an impressive paper in Nature Neuroscience 2 years ago now that showed the first use of focused ultrasound outside of MRI to produce neuromodulation. So low intensity, not the temperature modulating, but low intensity pulsed ultrasound to actually stimulate or inhibit neuronal activity on a millimeter spatial scale. So being able to do that inside a scanner while doing real-time functional imaging, I think is a home run. So lots of potential.
Anyone else?
What conditions do you see this starting to impact? Are there various conditions that
Right.
we haven't talked about?
Yeah, so on the focused ultrasound front, there's essential tremor, which is still people know Parkinson's better, but actually essential tremor is more common movement disorder. Actually Health Canada has given approval for the treatment of essential tremor with this technology just this past year. But tremor-dominant Parkinson's disease is another indication for it. Epilepsy, any of the types of neurosurgery where you're attempting to remove or destroy tissue, you could imagine. Right now, we're kind of limited to a central area of the brain, just because trying to focus the ultrasound too near the skull is yet an unsolved problem. But so there's a whole range of other neurosurgical. There's functional neurosurgery in certain psychiatric conditions, severe intractable depression, chronic some types of pain, OCD, obsessive compulsive disorders. There are a series of other ones. And then once you get into some of these other indications, you look at brain tumors, one of the problems with a lot of drug development for neurological applications is it's hard to get the drugs into the tissue. In the capillaries in our brain, there's something called this blood-brain barrier that actually blocks most large molecules from entering the brain. It's said that as many as 90% of drugs developed have trouble crossing from the blood into the brain. So the ability to be able to open that blood-brain barrier non-destructively to deliver drugs or remove unwanted products and have it close again without causing effect is potentially huge for all kinds of brain tumors and glioblastomas. Gliomas are the ones we're starting.
Other questions? I have one. The video, of course, was quite impressive.
Yeah, those are.
Impact of that on that patient's
Yep.
immediate condition. Is that a lasting effect?
Right. So, I mean, I think the longest a patient has gone in the world so far is around 4 years or 5 years from the earliest ones that were done. And yeah, I mean, it's a lasting effect. And the thing is, there's actually a long history of that type of surgery, but it's normally done. So what you're doing there is you're heating and destroying a small bit of tissue in a particular structure in the thalamus in the brain, and that's been done for years. Where the procedure is typically to drill a hole in the skull and then insert a probe all the way through the brain tissue down to the target you want, and either heat it with radio frequency ablation or freeze it cryogenically, or you can have a tool, a leucotome, which is like a piece of wire that comes up that you can rotate and just cuts the tissue. I mean, so there's sort of a long history of doing that procedure called a thalamotomy, but now we're just doing it with a source that doesn't require us to actually break the skin. So I mean, we have every reason to think that it will persist as long as it has in other conditions, in other approaches, surgical approaches. Yeah. Right now, it's Toronto and Calgary, in Canada. It's in Boston, Virginia, Zurich, Israel. There's some other places, but Stanford in California. There's a number of places. Yeah. I think maybe 15 places worldwide, 15, 20. Pardon me, accessible to? Oh, yeah. I think like a lot of these things, it will spread. Montreal will be getting one in the next couple of years, and then it'll be regional, referral areas. We're already doing cases from BC, and so there will be catchment areas where patients travel for that procedure. And with time, as prices come down and expertise grows, I think it'll become more and more widely available. It's not excessively expensive technology. I mean, it's a couple $1 million bucks. It's not insane. Right. Depending on precisely where it is, I mean, first of all, for a lot of tumors outside the brain, surgical resection itself is not a problem. It's in the first line, and you're not worried about necessarily getting to the tumor, right? A lot of it with brain tumors is getting to it, particularly if it's in a very difficult location. How do you access it? And the other limitation on it now is also it's difficult to do something in close proximity to bone. So because of the problems of getting the ultrasound through the bone and trying to get sufficient energy to heat and destroy without heating the bone surrounding it. So as you get too close to certain areas of the brain, acoustic neuromas are a tough one, and then it can be tough. And right now, we can't do those.
Just wonder what's the difference between a
So the laser-based ablation destruction, neurosurgical stuff going on to date is all with insertion of a laser probe. So there's none of it that's looking to focus from outside, without causing any issues along the way. And yeah, I don't know of any developments on that. The only sort of similar thing that's been around for a while that actually I worked on decades ago is something called radiosurgery, where it's similar in that you use a large number of radioactive sources outside that are brought to focus on a small area. So in most radiation therapy for oncology, the treatment is actually done over many sessions where you're trying to get this, take advantage of the difference in biological response of the tumor versus the healthy tissue and so on. But radiosurgery is meant to have a single procedure or a very small number of procedures that focuses a very high intensity amount of radiation there. And that's been around for a while and it's being used. There may be radiosurgical systems in the province here, I don't know. Is there a gamma knife here or a cyber knife? Any of those things? Not that you know. But those aren't, they take, they'll take time to take. So you do the treatment and you'll see the radiation effects evolve over a week or two. Whereas this is instant. And it allows you also, the fact that you can heat a bit but not destroy and still you'll get a temporary. So, in these cases you know the target you're attempting to hit. You can't see it very well on MRI, but you know it's inside of this blob, about that big, and normally it's so many millimeters down, so many millimeters out. So you go in there and you heat it a little bit, and you go talk to the patient, and you ask them to. Sometimes they say, "Well, I felt tingling on my lip." You say, "Okay, all right, I'm too far back. I'll try and move ahead by a half a millimeter." And you try it again, and you go in. Did you feel anything? No. Okay, and so you can move this around, and you're seeing the effect right away. And then you go in, and you say, okay, let's heat now. So you heat it, maybe to 54 degrees, and then you go back in, and you see, oh, okay, his tremor stopped, or he holds his hand and it's stopped, turns it this way, and you see still a little twitching in the muscles. So you say, okay, I think we can get that. It's really sort of an interactive and real-time effect that allows you to do that. Whereas with yeah, right. Yeah. And sometimes you treat and you sort of reveal other things, like there's an underlying dystonia or another type of movement disorder. Once you've resolved the tremor dominance, you see, oh, there's actually this other problem as well that we really never diagnosed because it was such tremor dominant. Some of generally the patients going through this have terrible tremors. It really is debilitating. They haven't been able to drink a glass of water or button their shirt. One fellow said, "Oh, it's been 20 years since I've been able to hold a glass of water in my hand." So when you see them come out and take a drink of water for the first time and sign their name, they cry, and everyone. It's impressive. Yeah.
Thank you once again.
My pleasure. Thank you.
I should come over and accept a little gift.
Okay. Thank you very much.
Thank you for coming. And I'd like to call on Jeff Emberley to come forward and say a few words.
Thank you, Dr. Peters, and again, thank you, Dr. Pike. Just a little bit about who Paganel is, Professional Engineers and Geoscientists, Newfoundland and Labrador. One of the reasons we do this is to show people a little bit about who we are as an organization. We're a self-regulating licensing body, basically, for engineers and geoscientists. And part of what we do is, that's most of what we do is those things, but we also have outreach aspects to what we do, and we're really delighted to partner with MUN on these Speaking of Engineering series. They're a great way to for not only others but ourselves. I found this tremendously interesting, and I'm sure every one of us did as well. And it's great to see how technology, engineering technology, the breadth of engineering technology I'm an electrical engineer. I mean, this is very far removed from where I ever practiced. And to be able to appreciate the breadth and the depth of engineering and what it contributes. But, much more to what it's all about was that last video. The purpose of engineering is really to impact people and to benefit people, and that really brings it home. And I really appreciate that. So, again, thank you very much for that, and it's much appreciated, and I ask you to show your thanks once more. And to you, thanks very much for coming, by the way, to everybody. And we have some refreshments outside, and feel free to stay and socialize. And if you have any more questions for Dr. Pike, I'm sure he'd be only too pleased to answer them. Thank you very much.